Hi, do you have any clue what I'm holding in my hands? It's actually a cell culture flask. We use it to grow human cells and to do experiments with these cells. The cells grow at the bottom of the flask. Not here, you're not supposed to hold it like that, but like this, flat, with the biggest possible surface area. So the cells grow here. What kinds of cells? Well, almost any type of cell you can think of. Muscle cells, liver cells, brain cells, kidney cells, immune cells, intestinal cells, stem cells, healthy cells, but also cancer cells, anything. Basically, what we do is we grow the cells on the surface and add a certain amount of liquid called the medium that is full of nutrients and which allows the cells to grow. Well, the cells don't really grow, they don't get bigger, but they proliferate, they multiply, which means we get more and more cells. But we don't just want the cells to multiply, we want to do an actual experiment with these cells. And with cultured cells, you can do a lot of different things. Normally, when you plan to do an experiment, you move the cells from this type of flask to special dishes, like this one. It's called a 12-well plate because it contains 12 wells. And each of these 12 wells contain the same type of cells. And the beauty of this system is that you can add something different to each of these wells. For instance, my research group wants to know which genes are turned on or turned off in cells by specific fatty acids like palmitic acid, oleic acid, linoleic acid. So what we do is we add these fatty acids to the medium and then leave the cells in an incubator. And after a couple of hours or even a whole day, we can harvest the cells and then measure which genes have been turned on or turned off by each of the fatty acids that was tested. Of course, we also need a controlled treatment where we don't add anything. It's a very simple experiment, but it illustrates the key advantage of cultured cells, which is that you can perfectly control the conditions and you can change only one factor from one well to the next. And any difference that you observe between the cells in different wells must be due to that one factor. In my example, we added fatty acids, but in theory, you can add any compound you can think of. It all depends on the research question. For instance, imagine that we're interested in the effects of quercetin on the breakdown of fat in fat cells. And quercetin is a compound present in tea, onions, raspberries, and many other plant-based foods. And how would we do that? Well, we would have fat cells in a culture dish and then treat them with quercetin for a day or so and measure how much fat is released by the fat cells. Pretty straightforward. Now that you've heard a bit about the setup of a cell culture study, it's important to point out the main advantages and disadvantages. The main advantage of studies in cell culture is that you can gain a better understanding of the molecular mechanisms of a certain effect. And that's usually the reason why these studies are done. For example, if we want to know if the effect caused by quercetin occurs via a certain receptor, there are tools available to disable or delete the receptor in cells. And if the effect of quercetin is lost, we can conclude that the receptor is essential. Another key advantage of cultured cells is that you can vary the conditions very easily. We can use different concentrations of quercetin. We can do the treatment for different durations and study other compounds alongside quercetin. And finally, studies in cultured cells usually do not take very long and are relatively inexpensive compared to animal experiments or human studies. The key disadvantage is that studies in cultured cells are quite artificial. Normal cells don't grow on a dish or plate, they grow 3D. Normal cells are not swimming in culture medium, but are in their normal tissue environment where they can also interact with other cells. Most normal cells in the body, in fact, don't even multiply, or only to a limited extent. Another issue is that people often make the mistake to add compounds to the cells at concentrations that would never occur or are not feasible in a living organism. So altogether, it means that studies in cultured cells can be very valuable if used wisely and interpreted appropriately. For example, just because a certain compound present in avocado slows down the growth of cancer cells in a culture dish, doesn't mean that avocado can slow down tumor growth. Please realize that any compound is able to kill cultured cancer cells if used at a high enough concentration. The trick is to use a concentration that's biologically meaningful.